Hello, and thank you for joining me. I'm Ross Cummings, and I am a UK and European patent attorney, specialising in biotech at Gill Jennings and Every, a leading patent and trademark firm based in the UK and Munich. This is my second talk in the GJE Expert Talk series. In the first, I explained the biotech-specific laws at the European Patent Office and provided general guidance for patenting biotechnology inventions in Europe. In today's talk, I want to delve into this topic in a little more detail in relation to the hottest medical technology of the moment, cell and gene therapy. Most of the issues I'll discuss today apply more generally to all biotech and pharma inventions. But as you'll see, cell and gene therapies tend to accentuate some of the challenges. I must stress that in my experience, provided there is some novel aspect to your invention, there's always a strategy that will enable you to obtain patent protection. Now, I will start by taking you through some of the history of gene therapy. Then I'll give some financial context to the current cell and gene therapy market, and I will consider the current approaches used in the clinic. While I will mention cell therapy at various points, I will focus on patenting gene therapy in Europe, mainly due to time considerations. In part two of this expert talk, I will look at the general trends and then go into particular challenges with this technology, how they may be addressed, and the various opportunities that there are to protect investment in this field using patents and other rights. In part three of this expert talk, I will go through some specific case studies and then finish by providing some practical take-home messages that gene therapy companies need to keep front of mind to ensure that an effective IP strategy is developed. Now, cell and gene therapies are the first real personalized medicines. Broadly speaking, these technologies take advantage of our knowledge of immunology, virology, the human genome, and genetic engineering to treat disease using nucleic acid and cells, rather than traditional small molecules or even polypeptides. In cell therapy, cells are harvested from the patient, genetically modified, and then returned to the patient. A great example of this is the booming CAR T industry, where the patient's own T lymphocytes are modified to express novel T cell receptors to target the T cells to the patient's own cancer cells. Now, this essentially awakens the patient's immune system to the cancer and has led to very long remission times, dare I say it, cures, in some circumstances for some patients' cancer. There are several approved CAR T therapies on the market and a strong pipeline of new ones coming through. There are lots of similarities between cell therapy and gene therapy in terms of the materials and techniques used. However, there is a subtle difference. Cell therapy typically involves enhancing the patient's immune system, whereas gene therapy seeks to restore a function that was absent due to an inherited genetic abnormality or was lost through genetic damage. As an interesting aside, I saw that recently Sanofi's announced their $160 million purchase of Tidal Therapeutics, who are seeking to develop cell therapies using messenger RNA to reprogram immune cells in vivo. This is an interesting blend of cell and gene therapy techniques that shows how quickly the field is moving. So in classic gene therapy, a gene is delivered to the patient's cells to either restore or compensate for a lack of functionality caused by a mutated and non-functioning gene, also called gene augmentation therapy. This can be effective in addressing so-called monogenic disorders, where a single gene defect is the cause of the disease. A good example of this is Novartis's Zolgensma, which is a treatment for spinal muscular atrophy, and one of only two gene therapies currently approved for medical use. The other is Roche's Luxturna, which is a treatment for monogenic retinal dystrophy. Gene therapy could also be used to silence a faulty gene, for example, suppressing an oncogene. Many conditions are caused by multiple genetic abnormalities, and this makes therapy more difficult. Nevertheless, it is possible to deliver more than one gene as necessary. Although with some vectors, there are space limitations for genetic payload that have to be addressed. This multi-gene approach is actually being taken by Cambridge-based Cuthera, who were acquired by Astellas in 2018 for £85 million and are developing treatments for glaucoma. In their gene therapy, genes that encode proteins that compensate for the lost function are introduced, rather than normal copies of the genes that are actually absent. Gene therapy was a hot topic of research in the 1980s and 1990s. The first human trial of a gene therapy was conducted in 1990 in a patient with severe combined immunodeficiency. However, an incident in 1999 sadly led to the death of a trial participant who had an adverse reaction to an adenoviral vector used to deliver a gene to treat ornithine transcarbamylase deficiency. This was followed by other clinical setbacks in the couple of years that followed that led to a steep decline in investment in the field. This slowed progress considerably, but in the background, many technological advancements continue to be made, particularly in an academic environment, such as universities. It's a different story now, and in the last decade has seen a dramatic increase in investment in cell and gene therapies companies who increasingly find themselves in top 10 tables for biotech M&A deals and private financing rounds. This has been possible in part due to the dramatic improvement in the safety of gene delivery to cells. By one count, nearly 300 experimental gene-based therapies are in clinical testing at the moment. The US FDA expects to be approving 10 to 20 cell and gene therapy products per year by 2025. A number of new life science funds have actually been set up in the last year. Most are agnostic in terms of therapeutic focus, but appear to instead be focused on investing in oncology and cell and gene therapy. Even in 2020, a year marked by the shutdown of much of the economy, there were very significant investments in cell and gene therapy companies. For example, $700 million raised by Sana Biotechnology in Washington, $493 million raised by Lyell Immunopharma in California, and Astellas' $3 billion acquisition of Audentes Therapeutics. Now, the action was not only over the pond in the US. The UK has a strong presence in these technologies, and UK-based companies have seen a significant investment. For example, Immunocore's $130 million investment, Freeline Therapeutics' $120 million Series C, and most recently, Gyroscope Therapeutics' $148 million Series C. It is estimated that by 2035, the UK's cell and gene therapy industry will be worth in the region of £10 billion. Cell and gene therapies are highly disruptive to the well-established and commercially successful pharmaceutical manufacturing, distribution, and pricing strategies. This is even before you consider that these therapies have the potential to be curative with just one treatment. With a healthcare system set up around chronic treatment, curative medicine will mean patients may not need to return for more medicine. Combine this with the small populations of patients with particular genetic disorders, and you need a new pricing strategy that the industry is only just starting to figure out with healthcare providers. In 2019, the NHS agreed to make Roche's Luxturna available to UK patients, which costs over $600,000 per dose at full price. Last month, the NHS also struck a deal with Novartis to make Zolgensma available, which has a reported list price of £1.79 million per dose. These price tags are hard to fathom for patients and healthcare providers alike. However, there are a growing number of gene therapy companies moving away from rare diseases into more prevalent conditions. For example, Cario Therapeutics has raised $80.5 million in Series A financing last year to advance their therapies for metabolic diseases, including diabetes, obesity, and Alzheimer's disease. Perhaps these approaches will lead to reduced costs for orphan conditions as technical and manufacturing issues are addressed. Now, Luxturna and Zolgensma, as I've mentioned, are the only two approved gene therapy products thus far in the US and Europe. But there was supposed to be a third by now, at least in the US, so-called Roctavian by BioMarin, which is a treatment for haemophilia. The expected approval was recently pushed back by a couple of years as the FDA wanted to see more data showing long-term effects. This hit their share price considerably, and maybe a sign of things to come. Longevity of treatment is a real issue that regulators are concerned about, and it's going to be one of the biggest challenges for the gene therapy market, particularly as repeat dosing may not be possible in a lot of patients if they show immune reactions to the vector or transgene product. It's also possible that gene therapy, as we know it, will be replaced with potentially more accurate gene editing technologies like CRISPR-Cas9. These are still early in development, though, and it is likely that the current rapid ascent of gene therapy is way ahead of it first. Also, Technologies developed for gene therapy, such as new vectors and manufacturing techniques, will likely also be applicable to gene editing technologies, where the only difference is the nucleic acid package, as it were, being delivered. So any work done to address the problems with gene therapy products should have broad applicability to future projects and products. And broadly speaking, there are two gene therapy methods, in vivo and ex vivo. In vivo gene therapy, the transgene is introduced into the body directly, packaged in a vector. In ex vivo therapy, a cell sample is taken from the patient, cultured, and then modified in the lab with the introduction of a transgene. Modified cell expressing the transgene is then returned to the patient to carry out the necessary function. One advantage here is that you can test there is a transgene being expressed in the cells before returning them to the patient. There are pros and cons to each technique, though, and ex vivo gene therapy is more similar to cell therapies like CAR T. Due to the differences in where the transduction takes place and how the reagents and samples are used, the patterns and strategies that apply to each method can differ in Europe, depending on the specifics of the invention in question. The vectors used in gene therapy are usually viruses, but not non-viral vectors have also been trialed. Viruses are naturally effective at gene delivery and can provide stable expression of transgenes, as this is their natural modus operandi. However, they do have certain drawbacks and limitations. Chief among these are the potential to elicit an immune response in the host, and integrating viruses have the potential to activate oncogenes. Viral vectors are used as vaccines as well, for example, the recent Oxford AstraZeneca COVID-19 vaccine. In vaccines, the host immune response is useful as an adjuvant, but if this happens in gene therapy, it can render that patient ineligible for future gene therapy, and in some conditions, significantly complicate future administration of conventional medicines, for example, clotting factors for haemophilia. This is because the patient can actually develop antibodies to the transgene, potentially worsening their condition and making it difficult to deliver that protein at a later time. And non-viral vectors such as liposome-coated plasmids address some of the drawbacks of viral vectors, but have been less effective in gene therapy, often due to only transient expression of the transgene. Most commonly used viral vectors are adenoviruses, adeno-associated viruses, and retroviruses, particularly lentiviruses. The industry is mostly settled on adeno-associated viruses for in vivo gene therapy and lentiviruses for ex vivo gene therapies. Adeno-associated viruses are small viruses that infect humans but are not known to cause disease. Both approved gene therapy products use adeno-associated viruses for in vivo gene therapy, as do many of the products in the development pipeline. This class of viruses has been used in gene therapy trials for over 30 years now and is still the mainstay of gene therapy. Progress in development of new vectors has been disappointingly slow, perhaps due to reduced investment after the earlier trials. Nevertheless, anecdotally, there does appear to be a resurgence in companies working on vector design and improved methods for gene delivery. The number of gene therapy patents filed and granted in Europe over the last 20 years has actually remained fairly consistent. No major increases or decreases. This might suggest a maturing field, but I think that we're only at the beginning of some rapid technological advances, particularly considering the significant investment that's recently gone into this field. One might expect the field to eventually move in the direction of non-viral delivery, but at present, ongoing trials do not show much evidence of this. At the end of the day, it will be the therapies that work that prevail. Now, in part two of this expert tour, I'm going to discuss some of the challenges to patenting gene therapies in Europe and strategies for overcoming these. And in part three, I'll discuss some specific case studies that illustrate these issues. In the meantime, if you would like to ask any questions, then please get in contact. Here are the details. Thank you very much.